Good evening. I'm Dr. Jennifer Cumston, the Dean of STEM at the College of DuPage. It's my pleasure to announce Dr. Trachette L. Jackson tonight. We are going to have her speak, and afterwards there will be an opportunity for question and answer, so when that happens, please wait for the microphone because we will be recording. Dr. Trachette Jackson earned her PhD in Applied Mathematics in 1998 from the University of Washington. She conducted postdoctoral research at the Institute for Mathematics and its Applications and at Duke University. She is currently a professor of mathematics at the University of Michigan. Dr. Jackson specializes in computational cancer research or mathematical oncology. With an eye towards addressing critical challenges associated with cancer therapeutics, much of Dr. Jackson's research is aimed at developing multi-scale mathematical models that are designed to optimize the use of anti-cancer agents that specifically target active molecular pathways that cancer cells use to promote their growth and survival. Dr. Jackson is an award-winning educator and scholar who has been honored for her accomplishments in both areas. In 2003, she became the second African-American woman to receive the prestigious Alfred P. Sloan Research Award in Mathematics. In 2005, she received the James S. McDonald 21st Century Scientist Award. In 2008, Diverse Magazine honored her as one of the year's emerging scholars. In 2010, she received the Blackwell Tapia Prize, which biannually recognizes a mathematician for both their research achievements and for their contributions addressing diversity in mathematics. Dr. Jackson has built her career on collaborative research and educational activities that can cut across traditional disciplinary boundaries and envisions that this type of team science will eventually change the face of cancer research. Please join me in welcoming Dr. Jackson.
Thank you. Good evening. It is a real pleasure and honor to be here to tell you a little bit about my work this afternoon. It's such a beautiful fall afternoon. It is really nice to be away from Ann Arbor a little bit and to get to share my work. I want to thank you for the invitation to join you and for that wonderful introduction that had all too many dates. I feel really old. Thank you. No, it was wonderful. Thank you. So I am an applied mathematician, and I work in the field of mathematical oncology, a relatively new branch of applied mathematics. And so I want to talk to you a little bit about my work in developing quantitative ways to help the design and optimization of cancer therapies. But I wanted to start with this article that appeared in Time magazine a few years back because they had this really interesting quote about what cancer is. And they called it, and so this is a Time magazine writer, so you know they're very good and descriptive. So they called cancer an intricate and potentially lethal collaboration of genes gone awry, of growth inhibitors gone missing, of hormones and epigenomes changing, and rogue cells breaking free. It works as a great armed force attacking by the equivalent of air and land and sea in stealth. And we think we're going to take it out with what? A lab-coated sniper? So this was Time magazine's description of what cancer is, what it does to our bodies, and then they ended with this question: what are we going to do about it? It's one of the most formidable diseases that we're facing in the world. And we need to come up with ways of doing better when we're treating cancer. So the purpose of them sort of bringing that question to mind was to say, cancer really isn't one disease. It's the name given to over 200 diseases. So there's no way you can expect that there will be one hero, one person that's going to come up with something to cure what we call cancer. Right? There's not going to be one hero, but there are going to be many heroes, right? So they were saying what we really need to begin to focus on is a team-based, cross-disciplinary approach to fighting cancer. That's what's going to upend tradition and deliver results faster. Now, in the Time magazine article, they mentioned a lot of team players. So they mentioned, of course, biological scientists, oncologists, pathologists, and then they said engineers and informaticists, and they forgot mathematical modelers. So I had to add us in there. There's me, right there. No, so we, like I said, it's a brand new field. We're just starting to crack the surface of what we as mathematicians can do in this field. And we're starting to get a little press, but we're still left out of Time magazine. But that's okay. So although cancer is 200 plus different diseases, most solid tumors have similar features. And so I want to go through a few of those, what we know about how cancers originate. So most cancers originate as normal cells that have acquired mutations over their lifetime of dividing, right? And those mutations cause those cells to transform. They transform into cells that no longer listen to the body's cues to stop dividing, no longer listen when the body says it's time to die, no longer listen when the body says it's time to be still, they want to move around. All of those things they begin to ignore, and they form a tumor. Another key feature of most solid tumors is this thing called angiogenesis. And that just means the formation of new blood vessels. So when we have these transformed cells that start to grow and make a little piece of tissue that's the beginning of a tumor, it can only get so big based on the amount of blood vessels that are surrounding it. If that piece of new tissue wants to get bigger, it has to create new blood vessels of its own to feed it. So that's a critical point in cancer progression, is blood vessel formation. When cancer actually connects itself to our existing blood supply, tricks our body into making vessels that feed it instead of the normal tissues. So once it's connected to our blood supply, it can begin to enter our blood vessels and travel. So these are cancer cells that are traveling through our blood vessels and end up in new sites, and that's called metastasis. So spreading of cancer to other sites via the blood vessels that it tricked the body to make, right? So these are characteristics of the progression of cancer. And we know a little bit more, well, actually we know a lot more about the biology that goes on with each one of these steps today. We know a lot about it, but here's what the data looks like. Every time we get data about how tumors grow, even though there's all that complex biology involved, the data looks relatively simple. We get information about the size, sorry, I'm trying to see where my pointer is, the size of the tumor as a function of the time at which it's growing. And on all of these are different data sets showing how tumors might grow. And we see similar characteristics in this data. All of the data seems to have this particular shape. We can see slow growth initially, this ramped up fast growth, and then this slower growth and leveling off at some point in time. Right? So that's a characteristic of the data we see all the time. All of this complex biology sort of shrunk down into this one simple kind of mathematical picture or graph, right? So classical models of cancer, as you might expect, didn't make use of all that complex biology. They just said, okay, we've got this simple kind of graph. We can come up with a mathematical way of describing that simple kind of graph. So before I tell you the kinds of work that I do today, I wanted to take you back down memory lane and show you where this all started, where cancer modeling, where the mathematical application to cancer and tumor growth really sort of began for most of us who work on it today. So the simplest models and some of the first models of tumor growth we attribute to two historical figures. One is Ludwig von Bertalanffy, and the other one is Benjamin Gompertz. We're going to talk more about him because his model is probably the most famous, Gompertz's model of tumor growth. It's interesting, their stories are interesting because neither one of them were interested in cancer at all. But they both came up with mathematical models that have been successfully applied to vast amounts of tumor growth data. And people still use both of these models today, even though neither one were interested in that. So we're going to talk more about the Gompertz growth model. But before I do that, I want to just walk you through how we derive a mathematical model. How do we come up with this thing that I'm calling a model? The first thing we do is we start with a problem of interest. For us, it's going to be eventually be cancer growth. And then we're going to make some reasonable simplifying assumptions because the biology involved is so complex. We can't model everything. We're going to have to simplify our region of interest. And then what we're going to try to do is articulate that problem and write it down in words first. And then we're going to translate those words into mathematical expressions. So those words are going to end up being mathematical statements of balance or conservation laws. So that's how we do that. Let's just do an experiment here and try to develop a mathematical model together, at least the word equation. Let's develop that together. Let's model. So as modelers, we feel like we can model just about anything. So if we were going to develop a mathematical model for the rate of change of the people in this room, right? So we'd start with a problem of interest. I want to know how the number of people in this room is going to change over time. What are some of the key ingredients that we would put into that model? What does the number of people in this room depend on? Time. Time, absolutely. What else? Space, right? This room can only hold so many. Yes? Rate of change. The rate of change of those things, yes. What about those doors over there? So people can what? People can exit and people can enter. Right. So when I wrote down this little word equation, I was being really thorough. Hopefully nobody's going to give birth in here during the next 35 minutes. And certainly, hopefully, no one's going to die. But we have to include those things as possible contributors to the number of people in this room, right? And then those things that you guys just said, entry and exit, which all depend on time, and there's a limit in terms of space. So we would write down this word equation, and then our real challenge would be to put mathematical formulas to what the entry rate would be, what the departure rate would be, and all of those kinds of things. So that's the main idea when we're talking about mathematical modeling. Now let's get back to Gompertz and see what he did. Okay, so his growth law, I told you, he was not even interested in cancer at all. He was interested in death. Human death. He was interested in mortality. He wanted to simplify the ways of calculating how many people are still alive in certain age brackets, right? So he was saying how many people are still living at age X. We're looking at specific age ranges, and can we predict the rate at which these people die within certain age ranges? So he did sort of the same thing. The rate of change of the number of living people in a certain age bracket is equal to the rate that they enter that age, the rate that they turn, say we're looking from 20 to 40. So the rate that people turn 20, right? And then the rate that they die. So that's how they leave that age bracket, or that's how they leave with death, enter and exit. So what he said was he wanted to translate something like that into mathematical equations. So what did he use to do that? Well, he looked at what in Britain they had back then life tables. So he looked at life tables to figure out what patterns there were in the data so that he could change those words into equations. He found that there were two different patterns that he saw, right? So first pattern, sometimes what he saw was that for certain age ranges, the death rate did not change with age. It was constant with age, right? Death rate didn't change. So he translated these words into equations based on this pattern of the data. So any calculus students in here? Anybody who's had calculus? Lots of people who've had calculus, you know that the symbols that we use for rate of change have to do with the derivative. So this is just the derivative, the rate of change, and that's just the mathematical symbol that we put to those words. And then he said maybe there's a constant entry to that bracket, and then there's some deaths with a constant rate d. Okay? So if you were to solve that equation, you would get the number of living people just declines exponentially. So that's an option. But the second pattern that he saw is the one that became the most useful pattern. So the second pattern was that in some cases, when he looked at this data, he actually found that the death rate increases with age. It's kind of intuitive. You would think that that would be true, at least for some age ranges. So he wrote down a form of increased death with age, right? So what happens to the number of living people if the death rate is increasing sort of exponentially with age? What do you think? Before, it just decayed exponentially, right? Number of living people just went down. Now, in this case, the number of living people starts off with an exponential decay. So that's the blue curve, starts off the same as the red curve, which is exponential decay, but eventually it's much faster than exponential. So this is the concept that ended up transforming the way people modeled tumor growth, believe it or not. This faster than exponential change in a population. So how do we get to tumor growth from that? The same idea, just flip-flopped a little bit. So now we're interested in cancer. So they took Gompertz's idea and said the rate of change of the number of cancer cells, right, is going to be how fast they proliferate and how fast they die. But what they said was that based on this data, just like he looked at data, these early modelers looked at data and said, the net rate of growth seems to be slowing down or decreasing as time goes on. So they wrote down that the net rate of growth should decrease as time goes on. So the same idea that Gompertz had for increasing death rate, they said was a decreasing growth rate. Same thing, just kind of flip-flopped. So this idea became the Gompertz growth model for cancer growth. Let's just take a closer look at so this is the same picture I showed earlier, but what it really shows is the diamonds and squares are the actual data for the growth of these tumors in mice, and then the solid lines are the Gompertz growth model. So the dashed lines, are the Gompertz growth model. Look at how well it matches that data, right? Problem with that is this graph compares several other mathematical formulas that have this slow initial growth rate, this rapid rise, and then this leveling off. Lots of models do that. So why is Gompertz the best one? Became the question, right? So there were lots of criticisms because any mathematical formula that gives you that kind of S-shaped curve, slow, fast, slow, fits this kind of data, right? And with the Gompertz model, we have no idea what's causing this slowdown in the net growth. There was none of all of that intricate biology that I showed you in the third slide, right? None of that was included to come up with those mathematical equations. So today, what we do is we improve on that because we want more than this, right? We want more than something that doesn't explain all the biology we know. Right? There's been such a boom in our understanding of the biological processes. We have to make use of that information, right? And we have to go beyond these simple general descriptions of the data, right? We want to get that out of the model, not necessarily use it to feed into the model. So that's where we are today, and that's where my work kind of begins, is with what we call mechanistic modeling. So by mechanistic modeling, I want to put in some of the structure from the underlying biology, some of the known things about the molecular and cellular processes that cause tumors to grow and that cause the cells to die into these models. And that way we can use them to predict better how treatments will work. So we are trying today, me and a whole slew of other people, are building these mechanistic models of tumor growth. So this is where I kind of began. So the title of my talk, of my work that I'm going to present today is Traditional and Targeted Therapies Against Cancer Stem Cell-Driven Tumor Growth. So I'm going to explain all of those words. And I'm going to start with the traditional and targeted therapies first. What do I mean by that? What are some traditional therapies for cancer? Chemotherapy. I heard radiation, all of those kinds of things. Surgery, right? One of the first lines of defense is surgery, chemo, radiation, Immunotherapy is still pretty traditional. It's like using your body's own immune cells to boosting those cells and their response to the cancer. Hormone therapies are pretty traditional. So what are targeted therapies? What do I mean by that? Well, for targeted therapies, what we're really looking at is designing drugs that interfere with specific things about a specific cancer. It could be specific cells that are made up in that cancer is composed of. It could be specific enzymes or proteins that these cancer cells are producing, or molecules that are necessary for the tumor to grow. So the primary goal of these targeted approaches is to use everything we know about the biology of a particular cancer to fight it with more precision. And if we can do that, hopefully we can eliminate some of the harmful side effects that some of those traditional therapies are known for, especially chemo. Okay, so I've explained traditional and targeted therapies. The next few words were cancer stem cell driven tumor growth. So what do I mean by cancer stem cells? So cancer stem cells, CSCs, I'm probably going to call them for the next few minutes, have been identified in a number of cancers as the driving cell type within the tumor, right? So they drive the growth of tumors, and there's some special characteristics of these cancer stem cells. They're typically only a small fraction of the cancer, so just a small number of them, but they drive the whole process. They have the ability to self-renew, and what I mean by that is that they can make copies of themselves, right? Exact replicas of themselves, an unlimited number of times. So most cells divide a certain number of times, and then they enter something called senescence, and they stop dividing, and they know when to die, right? These stem cells are able to divide an unlimited number of times. They are tumorigenic. By that I mean they are able to differentiate into all other cell types in the tumor. And they're often the ones that are difficult to treat. They're such a small population of the tumor, but they're also the hardiest in the sense that they're the ones that are resistant to many of the drug therapies, the chemotherapies. They can resist cell stress, which normally tells a cell to die. And they can also live through stressful things like radiation and things like that. So they're the hardest to kill. So this small population of cells is what we want to try to target because they drive tumor growth and they can create new tumors. So because cancers, several cancers are made up of these cancer stem cells, what that means is a tumor is not a collection of one type of cell. There's a lot of diversity. Every cell is different within the tumor. There's populations of different cells within a tumor. So we can have these CSCs, which are the red ones, and then there can be all these other cell types that are derived from these cancer stem cells, all populating the tumor. Okay, so it's important to remember that there's all these different classes of cells. We want to target this one little small population in hopes of killing the major driver of the tumor growth. That's the goal. Okay, so what does this have to do? What implications does this have for therapy? Well, traditional therapies, conventional therapies, the ones we listed first, well, if you give those therapies, here's the tumor. It has, cancer stem cells as a small population of them. You give those traditional therapies and you kill off all of these cells that cannot form a new tumor and cannot divide unlimited number of times. And what do you leave behind? You leave the stems, the driver, the worst cell possible is left behind, right? That's the cell who can start dividing again, that also can generate an entire new tumor. And eventually, all of that cancer recurs, maybe even in another location. If you had a targeted therapy that didn't really care so much about these cells that are going to die anyway, they divide a certain number of times then die, but can actually pinpoint these cancer stem cells, you may not see an initial shrinkage of the tumor because you've only killed a very small fraction of the cancer. But what would eventually happen as these cells die is that you would not get the regression. That's the theory of how this cancer stem cell therapy should work, right? So that's what we want to investigate a little closer. Okay, so remember I'm trained in applied math. I've been talking a lot about biology, but that's not what I'm trained in. So I have to have collaborators. I have to work with actual medical doctors and biologists and pathologists in order to do the work that I do. And my collaborators are interested in head and neck cancers. These are cancers that don't get a lot of press, I would say, but they're the 6th most common cancer in the world. So, and they include things like larynx, tongue, oral, nasal, cancers in the head and neck region. Let's see, so most of them are head and neck squamous cell carcinomas. I'm going to call them HNSCC. You might hear me say that set of letters again here in the next 5 or 10 minutes. But the reason my collaborators are so interested in this, Michigan has a big center devoted to understanding this, understanding these cancers. The 5-year survival statistics haven't changed for 30 years. We've made no progress with these cancers. There have been no new FDA-approved drugs for the last 15 years. Again, we're not making any progress on this. Mostly chemotherapy is the cornerstone of treatment for these cancers, right? And they are stem cell-driven tumors. So maybe there's some room to push the needle a little bit if we looked at stem cell-targeted therapies for these cancers that we just haven't made any progress in for decades. So, as I said, chemotherapy is the mainstay, right? Chemotherapy is what is done when you have one of these cancers. You are going to get chemotherapy. And the type of chemotherapy most often prescribed is this chemotherapy drug called Cisplatin. I'm going to call it Cis, all right? Problem with Cisplatin, is that regrowth happens almost 50% of the time. You get this drug, and like we showed in that diagram, you're killing off a lot of the bulk cells, and you're going to get regrowth. Almost 50% of the time, the cancer comes back. The other problem with Cis is that so here's a schematic of letting a mouse has a tumor of this size, you treat it with Cisplatin, and then you watch what happens to the stem cells. Untreated mice have a small fraction of stem cells, as we said. After you give this treatment, which is supposed to cure the cancer, you increase the number of bad cells, right? So we increase the stem cell fraction after therapy. So now we have more drivers of the tumor and less of the regular cancer cells. So when you think about this fraction increasing, you could say, well, of course, you have decreased the total number of cells. You've killed off all the bulk cells. So if you decrease the denominator, you increase the number, right? That is true. However, evidence also shows that Cisplatin actually increases the division or the proliferation and self-renewal of these stem cells. So it's not only a denominator effect, it's a numerator effect. So if we're calculating the fraction, not only are we making the denominator smaller, we're making the numerator bigger. So those two things are very, very bad in terms of the outlook for that particular cancer. So what can we do about this? We have to target these stem cells because the drug that everybody gets increases the stem cell pool. So we need to know more about these stem cells. So remember, the title of the talk was using cancer discoveries with the aid of mathematical modeling. So here was one of the cancer discoveries. We had to learn more about these cancer stem cells. And it turns out that they live, so in this little schematic, the purple cells are the cancer stem cells. They live really close to blood vessels. And things that the blood vessels are doing are helping them maintain their special properties. So this is a schematic of it, and here's a real image of a blood vessel, and all these arrows are the stem cells living right near the blood vessels. So that was the first clue. They live near blood vessels, so they must be living there for a reason. They must be getting something from the blood vessel cells. And it turns out they are. So the blood vessel cells are producing a molecule that is helping them to stay alive in harsh circumstances and helping them self-renew when they shouldn't. So living near the blood vessels is actually helping them because of this molecule that the blood vessel cells are secreting, this molecule IL-6. Okay, so now that's our second clue. Stem cells live by blood vessels. Blood vessels are giving them IL-6. That's helping them stay alive. Maybe we can stop that. Maybe we can block this molecule that the blood vessels are supplying the stem cells, and then we can reduce that stem cell number. So that's what we want to do. We want to figure out how to stop this IL-6. It's floating around. It comes in contact with a stem cell and leads to increased survival and increased division, right? So our options are to trap it. What if we gave some drug that just swallowed it up, sequestered it, trapped it? That way it couldn't have that effect on the cancer stem cells. So that is an option, and there are drugs out there that do that. The other option is to give a competitor. What if we could design something that interacts with the stem cells better than IL-6, but does not lead to the increased survival, increased proliferation? And it turns out there was already a drug on the market for arthritis that does this. So the idea was to repurpose this drug that's a competitor for IL-6, an IL-6 antibody that would block this interaction with the stem cells. Okay, so that's what we're doing. Let's see if it works. So this IL-6 blocker, TCZ, here's what happens. In vitro means like in the lab, in the petri dish. In vivo means inside the body. So in both cases, if you don't treat, you've got your small percentage of stem cells. And if you do treat with this IL-6 blocker, you reduce the number of stem cells. So that's good. On its own, would it be an effective therapy? It doesn't look like it, because even though you're reducing it, you're not completely clearing those stem cells. But maybe combining therapies, combine that traditional cisplatin with this targeted therapy, maybe we can get some better results. That's the hope. So that's the motivation and the goal for a lot of the things that we do. So first, here's what we know, just to recap. We know that our traditional therapy, which was chemotherapy, cisplatin, and our targeted therapy, which blocks this molecule that the blood vessels are making, TCZ, this therapy, they affect our stem cells in opposing directions. Cisplatin, the chemotherapy, which is supposed to be helping you, actually increases the number of stem cells. The targeted therapy decreases the number of stem cells. So we want to use mathematical modeling to propose a way of combining these two things, because the hope is, right, if you look at this figure, here's our cancer with the stem cells making a small proportion of the cancer. Again, the conventional therapy kills all of these cells, leaving the stem cells, we get relapse, right? If we just kill the stem cells, right, we get a very slow regression. You're really not killing off the tumor very fast. You probably won't even think you've done anything for a long time, because it's just a small fraction. But if you go over to this branch and you combine those things, you get the combination of these two effects, maybe we can actually get somewhere with the treatment. So that's the idea. So my group has been developing mathematical models in the same vein of the one that we developed for the number of people in this room, just a more complicated version of that, that uses all of that complicated biology that we talked about in the third slide. So unlike the Gompertz model, we didn't just look at data and say, here's the trend. We said, here's what we know about all the biology. And let's put mathematical equations to as much of that as we can in order to be able to predict these things. So this is just a schematic. I'm not going to show you all the equations and all that stuff that go with it, but we look at things at different levels. So when in the introduction, the dean said multi-scale. What is meant by that is we're looking at different scales of the biological organization. So we look at the cellular scale, so the tumor cells interact with the blood vessel cells and they form blood vessels and all that stuff that feeds the tumor. So that's one scale of the population of the cells. And then we have the dynamics of binding of like the chemical mediators like the IL-6. And then that causes some intracellular signaling that tells these cells what to do. So our models kind of cut across all of these different scales in order to answer that particular question we were looking at, how do we best combine these therapies? Okay, so not showing you all the equations, sparing you a little bit of the math. Here's some of the outcomes that we're able to get. So we have some data. I like describing our experimental platform, actually. So my collaborators have discovered a way of growing human tumors with human blood vessels inside a mouse model system, and you get basically the same type of tumor you would get in a human for about the first 3 to 4 weeks. Human tumor cells comprised of blood vessels made up of human blood vessel cells. And so what they do is they implant human cancer cells and human endothelial cells onto this tiny little scaffold. It's about the size of your fingertip. And then they implant that into a mouse. And it begins to grow and feed and connect with mouse blood vessels so that blood is flowing, but the blood vessels themselves are made up of human cells. So with that, they're thinking they're getting as close to growing a tumor, a human tumor, in a setting that they can control. And so that's the kind of platform they use to generate the data that I'm showing you here. So what am I showing you here? Don't get overwhelmed. It's a picture of the tumor volume, so how big the tumor is, the days after the implantation of the little scaffold with the cells. And let's see, what are we first showing? So in black, we have untreated control. So the circles and squares are the data, and the black is the mathematical model. So our mathematical model actually fits the data pretty well for untreated. And then what we do is compare what our model would predict if we treat with just the targeted therapy first. So what if we give the targeted therapy to our mathematical model and then compare it to what they got when they gave that targeted therapy to the mice? And that's what these two green curves are. So these are treatments on 2 different days. Again, the squares and the circles are the data, and then the lines are the mathematical model. And we were really happy when we saw that our model was predicting the reduction in a similar fashion as what they saw with the data. And then we also wanted to look at the stem cell percentage. Again, black is what happens when you're not treating the tumor. You settle down to some small percentage, like 1.5% stem cells. And then if you give the targeted therapy, you reduce that significantly, right? That was the goal of the targeted therapy. So right now we're just training the model, calibrating it, making sure it predicts the data in the right way. And this is just for the targeted therapy. We have to look at the same thing for the traditional therapy, the cisplatin, the chemotherapy. Again, the black line shows the model prediction of the tumor growth, and then the data is in the circles and the squares and diamonds. Again, for cisplatin, we see the model is following right along with the data. And then for cisplatin, when we look at the stem cell percentage, for non-treated, you drop down to a small percentage, but as soon as you give the drug, you get this bump. So the drug was given 3 times, and every time the drug was given, you see this bump in the stem cell numbers. So cisplatin is increasing the number of stem cells every time you give it. So just like we expected it should. So then what happens if you combine these? So the way that they wanted to combine the therapies was to co-treat. Let's just give both drugs at the same time, see what happens. And here's what our model predicts and what the data predicts. Again, black line is the untreated case, and then the blue squares and circles are the data, and then the lines are our model. And we see, again, the mathematical model is predicting the data very well, right? Not only that, we predict that when you combine these therapies, you counteract the traditional therapy's effect on the stem cells. It was increasing the pool, which is horrible, right? But if you give this targeted treatment together with it, you not only decrease the pool, you decrease it to lower than what it would have been. So that's great. All of this is good. But so far, all we've done is match the data. What can we really use this model for? What is the real power of this mathematical model? The real power of the model is to be able to say, what if you don't co-treat? What if you treat in all kinds of different ratios and dosing strategies and schedules? What is the best one? How should we really give these drugs if we want to have the best results? These results are okay, but the tumor was actually still growing, right? We delayed the growth, but can we do better? So maybe what we can do is use the mathematical model to say something about dosing schedules. Instead of just guessing what dosing schedule works best, let's test it with the mathematical model. What we want to do is minimize the amount of the targeted therapy that's required by changing the timing or the ordering of the administration of the two drugs, while maximizing the amount of stem cell reduction. We want to get that number of driver cancer stem cells down as small as possible, right? So minimize the amount of targeted therapy for a fixed amount of traditional therapy while maximizing the stem cell reduction. Okay, so I'm going to show you some tables. They're going to look a little scary. Just walk with me through them. And I'm just trying to give you an idea of what we were able to show with the mathematical model. Okay, so walk with me through it. So first, let's start off with the gray. The gray represents when we give the targeted therapy. So we give it on week 0, week 1, and week 2 here. We give it week 0, week 1, week 2, and week 3 here, so forth and so on. So this is three weeks of targeted therapy. This is 4 weeks of targeted therapy. This is 5 weeks of targeted therapy. Now, everywhere you see the word cis means we've added the traditional therapy, cisplatin, along with it. So the baseline in yellow in each case shows what they wanted to do. They wanted to co-treat. So they gave cisplatin, cisplatin, cisplatin along with the targeted therapy as the baseline. What we tested was, what if you pre-treat? What if I give cisplatin first? What if I post-treat? What if I give it later? Does this make any difference? So what were we looking at to see, does it make a difference? How did we answer that question? Well, we looked at 3 things. We looked at a measure for the amount of targeted therapy needed. So that's just a measure of how much targeted therapy we needed. We looked at a measure in the middle of drug synergy. What I mean by that is just how well the drugs are working together. So we looked at a measure of that. And then, of course, we wanted to look at the stem cell reduction. So the induced stem cell reduction is that last column. We want that to be as high as possible. We want the synergy number to be as low as possible. And then we want the amount of drug to be as low as possible. So what we find, let's just look at this first block of 1 through 5, where we gave 3 doses of targeted along with 3 doses of cisplatin, and we either pre-treated, co-treated, or post-treated. Our synergy number, which we want to be as small as possible, is always bigger than 1. We definitely want it less than 1. Bigger than 1 means they're antagonistic drugs. They're not working together at all. Less than 1, and the closer to 0 you get, the more they're working together. None of those numbers are less than 1. Actually, you can look down all these columns. It didn't matter what we did. These drugs are not working together at all with these dosing strategies. And if you look at our last column for the stem cell reduction, it doesn't change that much, right? I think the smallest number we have is about 47. The biggest number we have is about 51%. So not that huge of a difference no matter how we give these therapies. And we tried giving more drug, giving less drug, pre-treating, co-treating, post-treating. So far, our mathematical results are saying these drugs are not compatible, right? But then we said, okay, let's look at some non-conventional dosing schedules. This is what the experimentalists wanted to do because this is the norm, right? We said, let's look at some non-traditional dosing schedules. Now, again, the gray means the same thing. We gave targeted therapy. And SIS, or CIS means that we gave the chemotherapy, but now we've spaced it out. We gave alternating doses, we skipped weeks. We're trying all these different dosing schedules to see how we can get the best results. And the first thing we saw was that by giving a week off is the first time we got a synergy index that was less than 1. So now those drugs were working together. So we kept playing around with that to see how we could get them working the best. Now look at this. This is still co-treating twice. So then we said, what if we never co-treat and we give a week off of cisplatin? Maybe that can help us. And then we got our lowest number for synergy, and we got the drugs working together. And look at how small our dose of targeted therapy is, right? So we've got a tiny little dose of targeted therapy, huge synergy going on, and we're still getting decent stem cell reduction. So these non-conventional dosing strategies turned out to work out much, much better than what the experimentalists were thinking should work out for these 2 drugs. So I'm just going to summarize here really quickly. What we do in general, this is one example, what we do is work to develop versatile mathematical frameworks that can operate across the biology to capture the mechanism of action of drugs, the mechanism of action of cancer cells and what they do in their environment, so that we can hopefully say something about how to design and to optimize new treatments that are being discovered in the effort to combat cancer. So that's kind of what we do. I want to highlight quickly my collaborators. I cannot do this on my own. I'm one little mathematician, so I need a lot of help. So I'm highlighting my experimental collaborators are Alex Pearson, he's at the University of Chicago, Jacques Noor, and Alex Okalea. She's a graduate student, and he is her mentor. He's my main collaborator at the University of Michigan. And this is my former graduate student who worked on a lot of this work, who went off to do a postdoc now at University of Chicago with our experimental collaborator. So graduate students, experimentalists, computational biologists, all working together to kind of generate these results.
At this time, we have some time for questions. So if anybody has a question, raise your hand. We'll bring you a mic so that everyone can hear your question. So we'll start up here.
Have you had a chance to test out the non-conventional schedule of treatment yet?
You know what?
You knew I would ask that, right?
Thank you for asking that. I normally have a slide that shows what our next step is. So our collaborators have set aside tumors grown, I think they have about 30 tumors grown in the same way, waiting for us to give them the go-ahead to test this. We haven't given them the go-ahead yet. We are still testing a few other combinations, but as soon as we say, here's what we really, really believe, they're ready to go to work and do the experimental validation tests on this model. We can't wait for that to happen. Thank you for asking.
Other questions?
Do you think these results will be applicable to other types of cancers as well?
That's a great question as well. Thank you. Yeah, I definitely think our approach is applicable. Will the results be exactly the same for other cancers? No. But it's very easy to adapt this framework to address how other cancers grow, and maybe you wouldn't treat with an IL-6 blocker. You might have to treat with a EGF blocker or something else for breast cancer. But I think the general framework is very versatile, and the details will change from cancer type to cancer type.
I've got a question about the mechanism that CSCs go through the body with or without the normal cancer cell. If you have a tumor, let's say in the breast, and it starts to metastasize, does the metastasis go with the CSC in it, or does the CSC break away, or what? When you say that it wants to find a blood vessel, it seems that if it's outside the lymphatic system and it gets in the blood vessel, the entire mass is going through the blood vessel. So how does the CSC pop away to get away from the chemical that you're using?
That is a good question. If I understand your question right about metastasis, so these cells develop hallmarks of cancer, and one of those is the motility mechanism to break away. You're right. Cancer cells normally want to stay in a close-knit group. As mutations acquire and as time goes on, cells develop the mechanism to move on their own, break away from the herd, and individual cells do enter either the lymph system and the vascular system and travel and start new tumors. So it doesn't have to be the whole mass that go, or even a big fraction of the mass that goes. Individual cells are able to do that.
So your chemotherapy can kill the cluster of regular cancer cells.
Yes.
But that CSC is floating by itself midstream, downstream in the lymphatic system, wherever it's going, and that chemical can't get at it. Is that the problem?
The chemical is just ineffective with CSCs. So CSCs have protective mechanisms. They just don't respond the same way to these traditional chemotherapies as normal cancer cells do. For example, if a chemotherapy is designed to kill a rapidly dividing cell, CSCs can enter quiescence and just stop dividing for a little bit, let that chemotherapy float on by, and then start up division again. So they have all these protective mechanisms to kind of evade what we're trying to do with chemotherapy.
One quick more question.
Yeah.
If you were to do a cell analysis of the standard cancer cell versus the CSCs, do you find the DNA is different? Are there markers in there that says this is one and this is another?
That is a really good question, and yes, there are markers. Are the markers great and perfect? No. But the marker that my collaborators are using, they have 2 markers, and when both of those are positive, they call it a stem cell, and these are 2 well-known markers that sort of signal stem cell-ness, stemness, but they're not perfect. They're not perfect markers, but there are some markers out there.
Could you talk a little bit about how long, what time frame has gone into setting up this model and preparing these? And you talked about they're ready to pull the trigger when you move forward.
Yeah, I'm just scared.
When you give them the go forward,
Yeah.
you know, what kind of time frame you expect that to take as well?
So the time frame for development of this whole entire framework has been about 10 years. It's been incremental. We had to start, so we use a strategy of isolation and integration. So we isolated each system, each of the biological pieces. We had to do a lot of experimental work to train and calibrate each of those pieces. Then we integrated them together so that we had the molecular piece, the cellular piece, the tissue level piece. So it took a long time to get to these final results here. But now that the framework is in place and we've got all of the modules or components integrated, the work is going much, much faster. So, and it won't take them long to do the experimental validation tests. They just have to treat the mice and wait a few, I think we go for a few months here in order to see if the results match what the model has predicted. Yep.
Do you use mostly differential equations to come up with models? And I think 95% of differential equations can't be solved with the methods they teach us in class. So how do you solve your models?
That's a great question. So yes, these are all ordinary differential equations based models. They're all highly nonlinear and highly coupled. So no, nothing you've learned in class is going to solve this system. We typically use numerical methods to get pictures of what the solution profiles are. However, there are some, because we have a lot of equations, there's no way we're going to solve them, right? But there are model reduction techniques out there that we could try to reduce our model to the simplest subsystem where we could do some of that pen and paper math, some of that fun, get your fingers dirty stuff that you guys learn in class.
All right, let's say they do they continue on with the testing, and you get to the point where you say, this is the sequence. This is what we think is the best sequence, and we're going to recommend it. Now, how do you translate that or get that from, I don't know the terminology, I'll say the theoretical space that you're working in, right? And how do you get it into like real world?
Into the bedside, yeah.
Right, so do you have to team up with a drug company? Do you have to team up? What do you do, or how do you get it to progress?
That's a great question. So say the perfect scenario happens and that we predict a dosing schedule for these 2 drugs. They're both already FDA approved. They both are already easily available. Cisplatin is the cornerstone already. TCZ is a drug that has been approved for arthritis and is now approved as a CSC blocker, stem cell blocker. So it's just figuring out how to give them together. So what I have to do is team up with, so right now I'm working with Dr. Noor. He is actually a basic scientist and a physician. So he actually has patients that he sees, and so is Dr. Pearson. So I'm working with the right people who can get it to bedside for trials. I don't know what all they'll have to do to convince someone to change the co-treatment that everyone thinks should work, that isn't. But yeah, I'm working with the right people who have access to patients. It's just a matter of them being able to try it.
So you said like a trial. So would those 2 doctors be able then to conduct, we always hear about trials.
Yes.
Gets in those trials. So that's
Yes.
what their next step would be.
That's exactly what their next step would be. They're anticipating writing a grant to the National Science Foundation to start a trial based on these results. And it's just a matter of getting that approval and then getting that approved at the hospital level to try it on patients. Yep.
Your data seem to show that the ratio, CSC ratio never went to 0.
That's correct.
So is there some lower threshold that, it so significantly reduces the reproduction of the tumors that you're effectively cured for your lifetime?
That's a good question, and I'm not sure what that number or percentage would be. I would have to ask one of the actual doctors. I feel comfortable answering, a lot of biological questions, but not everyone, and that's one I'm just not sure what that actual percentage or ratio would be.
How does the cancer survive in the petri dish without the blood vessels?
That's a great question too. So they are continuously fed. So in the dish, we make sure they have all the oxygen and nutrients that the blood vessels would normally give them. They're basically fed with those things artificially. So the growth is very different. And also the cells that we grow in a petri dish have been immortalized for decades, and so they just have very different growth properties than a primary tumor in a human that sort of had mutations and are specific to this one patient. Very different growth dynamics, and we have to really artificially keep those cells growing since they don't have a blood vessel system to do it. That was a great question.
Right here.
About 10 years ago, my mother-in-law, who lives in Madison, Wisconsin, was diagnosed with colon cancer, and she was treated with chemotherapy and survived and was
Wonderful. Yeah.
cured. At the same time, one of her brother-in-laws had colon cancer, and he lived in Corpus Christi, Texas, and was treated there, and he did not survive.
Oh.
My mother-in-law said something about her chemotherapy had some form of cherry extract in it,
Really?
and apparently it was developed there at the University of Wisconsin. I wondered if you had heard about
That's very
this.
interesting. I'm going to go look it up. I had not heard about that. A cherry extract. That's very interesting. And I assume your brother-in-law was on a different regimen or something and did not get that?
Yeah, he was on a different chemotherapy, and he did not survive.
Yeah. It's very interesting with cancer. When I talked a little bit about precision, it's hard to be precise because everybody's physiology is different. Everybody's makeup is totally different, right? And small differences in the physiology can make huge differences in how drugs interact with your cells. So trying to get patient-specific, person-specific precision medicine is the forefront. I mean, that is the future of cancer research. That's what we're trying to get to, so that we know what to do with each individual's physiology. We're not there yet, but hopefully.
Yeah, I'd like to, typically when I'm solving problems, I like to use multiple approaches and multiple models.
Yeah.
It seems like your model is looking at it from a reactive perspective, meaning once the cancer is already established.
Yeah.
Is there something on the other end where it's more proactive to prevent it from even occurring? Have you looked at that in your modeling?
That's a very good question. I have not looked at that, but there's a vast amount of experimental literature out there. It's been looked at by both from the biological perspective and from a modeling lens as well, looking at the sequence of mutations that happen and what sequence is necessary to transform a cell to cancer. How can you stop that sequence? Is that sequence a unique sequence? All of those kinds of things are being looked at. I just haven't ventured into that myself.
I've got to ask you one more question, if you don't mind.
Yes.
So the CSC cells are not receptive to chemotherapy. They've got some protective mechanism. But why is changing the regimen make them receptive? If they're not receptive, they're not receptive. So if you gave it every day, fine, they're still alive. If you gave it once every 3 weeks, what does that do to suddenly they make them receptive to the chemo?
That's actually a good question. So it's not making What we're doing is not actually making the CSCs receptive to chemo, right? It's the targeted therapy that's impacting our stem cells. So that's the TCZ. And all we're doing by spacing things out, so TCZ happens to have a very long half-life in the system, and cisplatin doesn't. It has a quick effect. By giving the cells a little more time to sit with the TCZ, giving it a break from cisplatin's onslaught, letting it sit with the TCZ, you're actually getting the stem cells with the targeted therapy a little bit more than you would if you keep onslaughting it with the cisplatin, which is boosting the stem cells. Yeah.
Question over here.
My question is pretty similar in terms of we have a huge investment in traditional chemotherapy. It seems like the future is in the targeted therapies.
So. Yeah.
Are you being kind of encouraged to work with the traditional chemotherapies at the expense of moving faster with the targeted therapies?
Not really. I think the future is targeted therapies, absolutely 100% for sure. It's a slow-moving process, and the norm is to, once you have a new targeted therapy, well, let's try it with what's existing. It's hard to let go or sunset something like chemotherapy, which has been a hallmark therapy forever, right? So it's hard to let go of that. So I think more of the norm is to try to incorporate these new strategies along with chemo instead of sort of eliminating the option of chemo. And I think that's probably a good first step. Eventually, we may only have targeted therapies as our cornerstone, but that's a while away, I think.
Do you foresee your targeted therapy modeling eventually can be extrapolated and superimposed onto other diseases? For example, diabetes mellitus, for example.
That is a great question. That is a really good question. I definitely think it's possible, and you just kind of blew my mind a little bit with that, because I definitely think it's possible. I just haven't had time to really explore it, but I definitely think we could do that. I definitely think we could do something like that.
Okay, I want to, I know there's still a couple questions out there. I want to go ahead and thank our speaker one more time. If you still have questions, please come up and we can answer them up here. But let's thank our speaker one more time.
Thank you all.